They said this was a round table, and they weren't kidding. It's sort of an oblong table. I really want to thank everybody for coming this morning. I want to thank all of our participants, as well as everybody who's here in the audience to talk to us in this important discussion of 21st century cures. And I really want to thank National Jewish for hosting us this morning. Every time we call, National Jewish steps up. So thank you very much, Dr. Downey and everybody else who's here. As all of you know, I've joined Congressman Fred Upton, who's the chairman of the Energy and Commerce Committee, which is the committee that has jurisdiction over all of healthcare research, biomedical research, and healthcare policy. And we've joined together in launching this 21st Century Cures initiative. When we started the initiative in May, we knew it would garner a lot of interest, but we've been pleasantly surprised at how much interest that we've gotten. So far, we've hosted 3 roundtables and 6 hearings in Washington, as well as posted 4 white papers on different topics that have allowed us to seek feedback and guidance on how we can positively impact biomedical research and innovation in this country. Over the congressional recess, we were just talking before we came in, members of the committee are holding additional roundtables just like this one across the country. We've had a couple in Florida, we've had one in Michigan, we've had one hosted by Chairman Joe Pitts, who's the chairman of the Health Subcommittee, and he was joined by the ranking member, Frank Pallone. And for those of you who are used to coming to events where it's a panel, either a congressional hearing or a panel discussion that people have, this is kind of a different format. And the reason is because we're not doing this so everybody talks and makes a little prepared statement and then talks at you. We're really doing it so we can have a dialogue among experts and begin to get evidence. We're talking about a lot of different areas within the biomedical research and innovation arenas. How do we more effectively incorporate the patient perspective? How do we examine the role of incentives, potentials in the areas of digital health, modernizing clinical trials, and personalized medicine, just a few. So today, the topic of this roundtable is going to be personalized medicine. And we have experts from across the spectrum here who are going to talk about this. As we all know, there's not one definition of personalized medicine, but the concept is about developing and utilizing treatments and cures that meet the needs of an individual patient. We're going to hear from important communities in this field, the patient perspective, academic and research perspectives, the administration and the agencies, and representatives from laboratory and diagnostic science space. We know that a lot of advancements in science and technology have been made that lead to the successful developments of treatments and cures. And we're also seeing that as we learn more about the science, we can target potential treatments and cures more effectively on impacted populations. I'm eager to hear more from the participants today about what personalized or precision medicine means to them, their organization, their industry, and their community. So everybody should have a brief bio of the participants in front of them, so I won't take the time to read through it, but let me introduce everybody. First of all, Dr. Eric Green is with us. He's the director of the National Human Genome Research Institute at the National Institutes of Health. And Dr. Green, I want to thank you for coming here to Denver to be with us today. And then somebody who I've worked a lot with on different issues, the director of the Center for Drug Evaluation and Research at the Food and Drug Administration, Dr. Janet Woodcock, who has also come from Washington to be with us today. And thank you, Dr. Woodcock, for being with us. The Director of Cystic Fibrosis Clinical Research at the Children's Hospital, Colorado, and the University of Colorado, Denver, Dr. Frank Curso. Thank you, Doctor, for being with us. The CEO of Biodesics, Dr. I'm sorry, I can call you Dr. Burnell. You should be, but I don't think you do have a doctor. But he's going to talk from the private sector perspective. The Director of the Personalized Medicine Initiative and Chair of the Department of Medicine at the University of Colorado, Dr. David Schwartz, who's not here. Okay. Oh, he was planning to attend, but he had a last-minute issue, so I see now where you wrote that on here. I'm like, wait, where is Dr. Schwartz? Dr. Theodorescu, who's the Director of the Cancer Center at the University of Colorado, and who I've worked with a lot over the years. Thank you for coming. And then the Executive Vice President of Academic Affairs and Professor of Medicine and Pediatrics, and our host for today, Dr. Downey. And again, thank you to you and to National Jewish for hosting us. And so our goal is to have a free-flowing conversation about personalized medicine. So as is the tradition of these hearings, or of these roundtables, I'm going to pose an opening question to Dr. Downey, and then I'm going to open it for everybody to talk. So the first question, and everybody can answer the question too, if they want to, or they can talk about different things. What is currently happening in the field of personalized or precision medicine, and what's the potential of personalized medicine to impact biomedical research and innovation in the future?
Thank you.
I think you'll have to move that close to you.
So I'd just like to begin defining a little bit the definition of personalized medicine so we're all on the same page.
Is that on?
I'm not sure.
It's on.
I'll just move it a little closer.
So I'd like to begin by just defining a little bit the definition of personalized medicine so we're all on the same page. And really what we're talking about is the use of all available information to assist in the diagnosis and then the most appropriate treatment for an individual patient. And the reality is that this has dated back to 2,000 years to Hippocrates, who used this exact same approach. And I think we've refined on that a bit. And we'll talk a little bit about some of the growing pains that we've had. But what we're really talking about today are the omics, sort of use of genomics, proteomics, and metabolomics to refine our ability to diagnose and treat patients. And I would say that personalized medicine is alive and well, but it's experiencing some growing pains, and it's got a really serious distal reality check that we need to do. So, one of the basic problems and that has spurred development of personalized medicine approach is the use of medications, drugs. And the fact of the matter is that for most drugs, less than 50% of patients who are treated with a given drug will respond positively, and in fact, a certain percentage will respond negatively, in fact, very negatively. And each year, over 100,000 North Americans die and 2 million are hospitalized because of adverse reactions to drugs. So that's a fact that we have to deal with, and the use of these technologies to predict, for example, response to medication or untoward effects is part and parcel of personalized medicine. That's a key. Another issue that I think is extremely important, and I know that both Frank and Dan will speak to this, is the ability to use advanced technology to predict response to therapy in cancer, for example, or in inherited diseases such as cystic fibrosis. And in fact, in both of these disorders, there are specific targeted therapies, molecular therapies, if you will, that have been introduced that are highly effective in small subsets of the population. So again, the personalized medicine approach can be used to identify those and establish those appropriate therapy. But now comes one of the reality checks. Some of these medications are incredibly expensive. In fact, Caftor, if I pronounce that correctly, between $280,000 and $300,000 a year. Some of the drugs for cancer, the EGFR inhibitors, can be hundreds of thousands of dollars a year. And no matter how good a drug is, if patients and the healthcare system can't afford it, it becomes less useful. And then lastly, I know that this is going to take a lot of discussion, some of the barriers that have been imposed in the last couple of years by regulatory CMS stopping paying for all molecular testing without any warning, sort of put a big roadblock. And then the FDA, in terms of appropriate setting of targets for approval, but some of those bars are so high that I think is creating a problem with financial viability. Can we really afford, as small or medium-sized institutions, to be in this innovative space if the bar is so high that we can't achieve it? So I'll stop there and say that's my view of personalized medicine in a nutshell.
Okay, thank you. Who else?
I'd be happy to say something.
Thank you.
Yeah, we at the FDA have been interested in personalized medicine for a long time, since even before 2000. And why, at least from where I sit, the Center for Drugs, we have to regulate all the drugs that people take. And as Dr. Downey just said, we get a lot of adverse events. We get a lot of drugs people say they don't work very well, but that's because they aren't targeted to the people who stand to benefit. And we don't understand why they're getting these bad side effects. In the last 14 years, since 2000, and the sequencing of human genome, we've learned a lot more. And we have actually tests that can weed people out who might stand to have a very severe side effect if they're tested. And we also have, as was said, there are various tests that can be done to pick the people who might respond. And so what we're seeing is much higher response rates. And in the face of that, it's much more tolerable to have side effects because you know you're going to get some benefit in that population from the drug. So the promise of this is tremendous for treating a wide variety of diseases. It is the medicines of the future. That's what we're going to see. We're going to see the science, and we're seeing it now, rapidly dissecting what causes disease, and then thinking about how we can intervene in that specific disease. It's like we were flying blind before. We were just empirically trying to treat. So from the drugs perspective, it's extraordinarily exciting time because we're on the verge, I think, of a therapeutic revolution where we're really going to be understanding the diseases that we treat. But it's a process and a transition. There's going to be a lot of growing pains. There are going to be a lot of issues that we have to overcome. But for the FDA, for drug regulation in particular, this, and I think for diagnostics as well, this is a tremendously exciting time. What we need to do, I think, is figure out how to get the promise of this without, and overcome these barriers. And my belief is that science is what will bring us out of the problems that we're having now. We may, can we use platform technologies that then we can look at more collectively and not have to validate each certain one? Can we get new trial systems in place so we can scientifically evaluate these things? I think there are scientific solutions if we have the will and vision that we can sort of midwife this new way of treating people and really make it work for everybody.
Everybody here. We'll have
So, Congresswoman DeGette, thank you very much for organizing this and your leadership in the 21st Century Cures. And I think the introduction that was given by Greg was excellent. I would like to add a couple of things. I think we really need to think very carefully about potentially putting forward a paradigm change of how we do things. And I think the way I envision this paradigm change is really to address really the core issue of how we do clinical investigation in this era of personalized medicine. Because what's happening is that we do these very expensive clinical trials. Most drugs take, cost about $2 billion, at $1 to $2 billion to go to market, 10 to 14 years. And I think if we really have to do that every time, we're not going to make an advance. And I'm speaking with obviously the optic of cancer, because there are many drugs, there are many targets, and we're just not going to have the bandwidth, nationally or internationally, to be able to do this. So I think we need to have a better paradigm. One of these things is that every trial, for example, should be cataloged, and at the molecular level, tissues should be examined, and these data sets should be available nationally. For example, We know from the cancer prototype, for the cancer phenotype, that a subset of patients may respond to a drug, and yet the trial may be negative overall. We need to have the molecular intelligence to be able to gather that information, not only from drug X, but drug XYZ that is similar in mechanism. And so I would love to see, for example, a national data repository where people can do big data mining to basically understand and essentially peel away the subsets of patients that respond. And hand in hand with that, you would have the idea that every targeted drug, I feel, should go into trial with a companion biomarker. So the markers and the drugs can be validated and their effectiveness assessed at the same time in the clinical trial. Because then, at the end of the clinical trial, not only will we know what subset of patients are affected, are most well treated by this, but we have a tool that then can be used to then apply this drug in clinical practice. And even if we have a success in 5% of patients, that's 5% of the onion that's been peeled, leaving 95% for the next wave of drugs. And I think that's how we're gonna really deal with the cancer problem, by peeling it 5% at a time potentially. But eventually it'll go down to 0.
I will agree with all the previous speakers that there is such a thing as wellness and molecular medicine. I want to talk about the person. In cystic fibrosis, we have to go over research opportunities basically at every patient account. And if we don't, the families will ask you. So you have to stay up on it. And we need to involve the families at every step of the way, and the patients. This implies a couple of things. I'm not just talking about counsels or approvals, or something, but actual face-to-face. It implies a registry, a patient registry, because even with personalized medicine, putting all the data together we're talking about, it's going to have some relationship with the condition. And so we need a national registry for each of these conditions. When we approach patients for approval for the studies, we have to have good data from the registry to tell them, number one, what can they expect for the treatment? What would be a good outcome? It takes a great deal of courage to agree to do a clinical trial. It takes even more courage if you sign for your child. So we have to know what the risks are, and we have to be able to tell individuals what the risks are, and that we're trying to control for them. And in cystic fibrosis, we do this through the clinical trials network, which was started by the Cystic Fibrosis Foundation years ago before we had molecules, or at least almost. So I think personalized medicine implies face-to-face contact, implies registries, it implies clinical trial networks, it implies biomarkers. And I think others will probably speak to the need for steady funding for the National Institutes of Health. And there was a tremendous amount of basic work done in cystic fibrosis through the NIH and the CF Foundation before our friends in industry take the baton. But the FDA has an awesome responsibility. I have trouble keeping up in CF. I have some of my patients' families are here can vouch to that. Okay. But the FDA has to deal with over 5,000 other rare diseases, plus the 2 or 3 conditions that we all have or will have. And so, again, I'd like to see anything we can do to strengthen the Food and Drug Administration. They helped us a tremendous amount this year. Part of this was Senator Bennett's work, I don't know if he's here, with the breakthrough designation.
I worked on that in the House.
Yes, I mean, we got that through very quickly. one reason we could do that is we had the clinical trials network, we had the patient registry, so we could go to the FDA and say, help us with this, what do we do? And they helped us. So I guess those are my major points. The last one is just that it takes a lot of collaboration. It takes collaboration from the families, from medical schools, from hospitals, from research institutes, from our partners in industry, and very much so from the federal government, especially with the NIH and the FDA. I'll stop here.
Sure.
You're in the middle of this.
I'm in the middle, so let me give a context here, because we're only about 10 or 15 minutes into this, and we have gone across a
Right.
very wide swath of the landscape. And every one of these points that have been made, every one of these areas are incredibly important. But I want to make sure people appreciate the context, because I don't want people to get lulled under a false sense of believing that some of these things are completely solved on the more proximal side, especially on the science and research side. Because I think it's really important to know what we have and what we don't have yet. Because we need to be very careful to manage expectations and also to give you the kind of information you are asking for to make sure that whatever Congress might decide to do adequately takes care of the entire landscape and not just one little piece of it. So, and I will admit my biases here. I graduated medical school and graduate school just a little over a quarter century ago, just when the Human Genome Project was getting out of the gates. I got to participate at it really as a newly minted physician and a scientist, and participated from day one. And now find myself, a quarter century later, leading the institute that was created by the U.S. Congress to lead the U.S. Human Genome Project, or the U.S.'s effort in the Human Genome Project. And what our focus is now, and I'm speaking on behalf of the whole field of genomics, is to capitalize on this incredible accomplishment and to be able to catalyze the things that you just heard some of the other speakers say, is to make all of these things just sort of move down very efficiently to think about better treatments, better diagnostics, and then, of course, to be able to personalize, of which genomics is one aspect of personalizing, but be candid, it's the one that's really catalyzing a lot of this because of what's happened over the last 11 years since the completion of the Human Genome Project. So, but I liked what Dr. Wincott said, because she said, remember the science, remember this is not all solved and just ready to deal with on the clinical side. You know, we finished sequencing the genome, human genome, about 11 years ago. We need to eventually completely understand what that code means. We're maybe about here. We need to be about here in that understanding. Meanwhile, we're getting it, we're figuring a lot out. Meanwhile, we recognize that information in each of our patients' genomes will give us clues as to what rare diseases they might be susceptible to, what common diseases they might be susceptible to, and when they get cancer, to open up the blueprint of those tumors, read that out and figure out the best way to treat the patient. We need to accomplish that. We had to sequence the genome not one time. We need to be able to sequence any patient's genome, any tumor's genome. But 11 years ago, it cost almost $1 billion to sequence a genome. But we had, through a major US government effort, a funding program, to be able to develop technologies to reduce the cost for sequencing a genome. Now it's almost down to $1,000. It's within striking distance of the diagnostic. And that is paradigm changing, which is what we call it. That is paradigm changing. Now, does that mean we know the cause of every rare disease? No. Does it mean that even for the rare diseases that we know, do we know all the right treatments? No. What about common diseases like diabetes and hypertension and asthma and Alzheimer's? Do we yet know? But oh my gosh, in the last 5 years, have we begun to learn the approaches to use to figure out what the differences are in our genomes that make us more susceptible to these common diseases? And then I won't even speak to cancer because we have a head of a cancer center here who basically say that genomics and these new DNA sequencing technologies, genome sequencing technologies, are changing the face of cancer research and hopefully eventually cancer treatment. But I want to go back to this idea that we're not there yet. And a key part of it is to make sure that we take advantage of these incredible opportunities, that we see enough good examples to know, we have enough good examples in rare diseases, we know what we need to do. We have a few examples in common disease, we know what we need to do, and in cancer it's very clear what we need to do. one of the things we need to do is to be able to push the accelerator really hard, but also to not have barriers in our way so that when this enterprise of research goes forward in personalized medicine, that there aren't obstacles that just impede progress. But we're not there yet, and I think that's one of the key things, is we need to think about the entire landscape, starting from those basic research activities and every other thing, every other person mentioned so far.
David, let me bring you in here because we're not going to let you get off for now. Dr. Green just mentioned barriers to finding cures, to getting it from the basic research to the actual cures. You've been involved for many, many years from the private sector in investing in companies that are trying to find diagnostics and cures and things like that. What regulatory barriers do you see that prevents private business from investing in these kinds of endeavors where the private sector can actually help bring these cures to the patients?
Should I?
Yeah, here's the mic.
So, yeah, I think we had a very wide-ranging discussion.
And any other
Talking for
comments?
an hour. The industry, thank you for inviting a voice for the industry. While we're primarily a diagnostic company, in the process of using our diagnostic tools, we've actually been able to find some of these subsets of patients that respond for drugs that didn't move forward, so we're able to start moving them forward. The omics thing that we talked about, whether it's genomics, proteomics, metabolomics, is really important. And in this era of personalized or precision medicine, and while it's incredibly important that the patient is at the center of that, and that the doctor is there with the patient, looking at the very big picture. In order for this to move forward, on the diagnostic side, we are being inundated with a tremendous number of markers, if you will, interesting genetic information, proteomic information. And first, diagnostics has always been sort of a second tier to drugs. So drugs get reimbursed based on what the drug company decides the price should be. And diagnostic gets reimbursed basically the cost of doing the diagnostic plus a little bit. That's kind of what people have decided is the right approach. So if we're to use diagnostics to its full effect, we have to kind of change that balance, because diagnostic companies can take even our existing armamentarium of generic drugs that were approved, in total populations, but we know subsets don't respond, and start using those drugs much more effectively. But in order to do that, we have to do what the FDA wants, which is phase 3 clinical trials where you're actually proving that the subset you found gets the benefit, the subset doesn't, has a better alternative. And that's very expensive. So we talked a bit about registries and getting patients involved and making all of this data available. I think patients have a big say in saying, if I'm going to be part of a trial and take a risk and put my tissue out there, my blood out there for you to look at, I want it to be more generally available for science to take it forward. And that's not something we're very good at all. So for us to get access to sample sets is extremely difficult. We have to go pay that or buy it, and often that's only something that takes us 4 to 5 years to get. So I think, and I think for us to get advantage of all of this data, we need a lot of patience, because it's really being able to look at all of that data, sort through it, and then be more precise. Because it's not so much one patient, but a group of patients that we identify that have a common set of characteristics. So there's reimbursement things, and we've made progress. We appreciate your support for some of the clinical lab reforms in the summer recently. That was very helpful. The devil's in the details, so we really want our representatives to be paying attention to how law gets translated into practice. And then on the FDA side, I think science is really important, and making sure the FDA has enough money to keep up with the science. It's hard for you to keep up. They have to deal with a much broader picture, and if they're not funded properly to address the issues they're going to take a safety approach first. And that's going to inhibit our ability to move this technology, put the accelerator on it. And so recently we have the LDT CLIA guidance, which will be coming out, kind of been announced. And we're hoping we're not fixing a problem that didn't exist by doing this. And it's, I know that we're having discussions with the agency. I think they're fruitful. But it's really important that we keep an open mind about how these new technologies are going to be brought to market. And we've traditionally had a pretty an open opportunity with LDTs, and I think most of the companies in our place are great actors. But just the threat of an LDT or a change in CMS will dry up funding for diagnostics. And we've certainly seen that in the last 5 years. Some venture capitalists that had great experience in the space, very committed to it because of the promise, just basically decided to quit funding it because the hurdles to get a product to market are keep going up, and then nobody wants to pay for it when it gets there. So anyway, that's kind of the little bit.
Yeah, I think all the problems you referred to are kind of interrelated, and partly diagnosis is the foundation of medicine, not treatment. And in this country, we kind of have it backwards. We rush to treat people, okay? But what personalized medicine is telling us is we have to understand, and the better we understand what they have, the better our treatments can be, both for safety and for working. So I think it's critical that we address these issues with diagnostics. They've been around for a long time, especially the reimbursement issue. And I think it's just really critical that we figure out a way to elevate diagnosis to where it belongs in kind of the sequence of events in treating anybody. And yeah, FDA is committed to move, provide additional guidance and so forth, and that will happen in a timely manner. But I think there's a bigger problem underlying this, which is kind of we put the cart before the horse. Dr. Dow.
So I really underscore your point. I think it's critical. I want to come back to something that Frank brought up. And if I go back, and I'm probably the same vintage, I remember moving to Toronto at the time that Lapchi and his group were just finishing sequencing and identifying sequencing CFTR gene. And I remember the great expectations that we found a gene, we're going to understand this disease, and we're going to have a cure in a few years. Well, 20-something years later, it's just starting to happen. So the first point is that we have to be willing to understand the science, to relate the genetics to the disease. This comes to your point about phenotype, clinical information. And the next point I'm going to make is that one of the great successes of the cystic fibrosis community is that they pooled their information. And it started out with the clinical diagnostics. I mean, many of the people who would be diagnosed with cystic fibrosis now wouldn't have been diagnosed 25 years ago because we didn't know the spectrum of manifestations. So one of the big impediments that I see right now is sharing of this information. We're all being driven, for better for worse, to electronic health records, meaningful use. It's that cart, that is way down the road. But you can't communicate between electronic health records very well. So phenotypic information, critical elements about the disease that we're treating, we can't share easily. And to me, that is an impediment that we have to be able to address. That information has to be able to flow, in an unidentifiable manner, so to speak, to a central repository where it can be interrogated and linked to the genomics or other roles. We don't really do that well right now.
I'd like to say something about that. Chris,
You want to look?
okay, this is really a passion of mine. I really think what cystic fibrosis did is really exemplary of what, on a very small scale, and you could get your arms around that population, what has to be done. Because I think we really lack a translational apparatus right now to take this science and actually instantiate it into treatments for people. We just have it's like the missing link in this chain. And you think of networks on a much larger scale. So what did they do? They linked all the clinical information about the patients to their genetic information and so forth. They held that together. They had all the patients available. They set up a network of centers of excellence that allowed iterative improvements while we were waiting for, more definitive therapies. And they kept iterating that and they invested in a pull to bring therapies into that network and evaluate them. I think that needs to be done on a much broader scale. We can't just do the science, we gotta do the science. We can't just do the science, we gotta do the clinical science too.
So we're hearing a theme, and I really want to emphasize it, because I know you're looking for things that the U.S. Congress can help with, and I think you can pull this together and actually come up with a very concrete one. CF is being highlighted as a success story where the community of families and patients, they collect the data, shared the data, and that accelerated the science. That's a small community, relatively. It's a rare disease, but it's highly illustrative. But now let's think about what we want to do beyond all of the rare diseases. Let's think about the common diseases. And the common diseases in particular are going to be far more complicated because we already know that the genomic contributions to complex diseases are complex. These are common diseases. And we know we have to do it at a much larger scale. And then we heard already some things about patient empowerment, and I heard from you wanting to pool data. One of the most concrete things we need to have happen is a shift in the mentality of the American people to believe that participating in biomedical research as research participants is a great thing to do. And the reason why, this is going to be going on internationally as well, because actually we need greater numbers than just what we can provide even in the US. But if we're going to tackle diseases like diabetes, hypertension, cardiovascular disease, just go right down the list, Alzheimer's, we're not going to just be dealing with thousands of patients that we have to study and have their genome sequenced and do other omics studies and get their electronic health records and have it all pooled and available. We have to do tens of thousands, probably hundreds of thousands. And then we need to think about ways to knock down barriers of why people are not participating and incentivizing them to do that. And I think there's some very concrete things that could potentially be done so that it almost is with pride that people want to help the enterprise of improving the health of humanity by participating in research.
Well, it's interesting because in our previous hearings and roundtables that we've had, a lot of our witnesses and experts have said that they think that, and actually I'd like to hear from some of you, Dan, about people who you have in clinical trials or people who you work with and treat. A lot of people have, including patient advocates, have said that they think the American public would be very, very willing to donate their genomics to
Ask the next generation. Ask all the teenagers who are walking around with iDevices and going on Facebook. They want to participate. They want to share. That's the group we should be really getting excited about this.
Right. And so my next question, this goes to something Frank was saying earlier too, though. How do you really encourage and foster those tissue banks and the genomics and all that, while at the same time protecting patient medical privacy? That's a key issue, I think.
So these are incredibly important issues and near and dear to my heart because we're thinking about them all the time, about bringing people together. And so I think the development, at least on the cancer front, of consortia that will bring together the data sets among several cancer centers. We have something like this in the works now. And then look at that in aggregate and do some of the things that I mentioned earlier. I think it was going to be really productive. But sometimes, just to quote a saying that we all know, is sometimes you have to spend money to make money. And the whole fiscal issues around personalized medicine, it may sound like it's science fiction, very expensive, doing genome sequencing, but sequencing a genome is no longer a very expensive proposition. I would argue strongly that, in fact, to really do to reap the benefits of this, we should be examining patients genetically, whether it's a tumor or benign conditions. We should examine them prospectively, because then not only the diagnostic biomarker, the companion biomarker aspect is enhanced, but all the diagnostic companies will go back to these large repositories that have been collected in patients in clinical trials, and they, I would propose, no longer have to do prospective clinical trials because the tissues have been acquired in a prospective way. And therefore they can look back at those tissues in a virtual prospective clinical trial, saving them enormous amount of money and enormous amount of time. The other real challenge when we do these multi-center trials, which are going to be progressively more common in cancer as we're dealing with smaller subsets of patients with defined genetic mutations in cancer. We're going to need a lot more institutions, because no one institution will have enough patients. What we're going to really need to do is grapple with the issue of IRBs, because the IRBs right now add a significant amount of time to the process. And if you imagine a clinical trial with a novel agent that targets 5% of patients with bladder cancer, then that's going to take probably 30 or 40 institutions to open that trial. We really need to have federal funding. Any trial that is federally funded should have central IRB, and that central IRB should be taken at face value at all the institutions. Because if we have that trial approved across 50 different institutions, well, we can all do the math and the time.
For some of the lay people in the audience, talk about how, if you do a clinical trial, how's that done with the IRBs?
So, want to talk about an IRB is?
Yeah, well, yeah, sorry.
So it's basically a board that approves the ethics of the clinical trial, essentially. Basically, to make sure that the subject is protected, the person that is involved in the clinical trial is protected, and that no risk, undue risk, it's always an equipoise between risk and reward, basically the patient is exposed to. So it's basically human protection, so we don't do things that are not appropriate. The problem is that every time we have a trial, a national trial, that national trial has to be approved at every institution that opens the trial. What I would, and I think I'm not the only one that many of us would like to see, is at least for federally approved trials. So these are trials that are done nationally that either are supported by a federal grant or part of a cooperative group. Those trials have been through exhaustive scientific review. The science is fine. The ethics is fine. They should be approved by a central IRB, and then that approval should basically be taken at face value at all the other sites. That would save us an enormous amount of time, an enormous amount of money, and more importantly, you will get really important things done fast because patients are dying.
No,
And we can save
Main Street, a great deal, and I would not limit it then to just federal funded studies, because there's no reason why the same ethics committee or IRB can't rule on a industry-sponsored study with the same degree of rigor and trust, and that would eliminate a huge cost upfront to getting a trial open.
You know, I just have to jump in. Some of you know, I've been working on IRB reform for many, many years. And it's so interesting because what I've been working on is that we have different standards throughout academia and private industry and even different definitions of the common rule and how do you use that. And so this only takes that debate to a higher level. It's been a really interesting and kind of frustrating effort on my part because every time I get one group of people to agree what the standards and definitions should be, somebody else disagrees. So maybe, David, what you can do is you can get together with Dan and everybody can work with me on how we can make this happen. Because I agree with you, it's a problem right now. And as we move more and more to the types of research that everybody here is talking about, it's only gonna get more, it's gonna be more and more of a barrier. Because different sectors and different types of institutions have different definitions.
And if I can add one, you know, I think the issue of the IRB is critical, and also it can be very well thought as a one unit when it comes together with the networking idea and the coalescing of data, because we have informatic systems and we have the ability through honest broker systems to basically anonymize data for people to use. This is not putting a man on the moon. This is actually, we can do this. We need just the infrastructure to basically say this is important, this is what we need to do in order to make advances. So it's not that we have to reinvent new technologies. We can do this now.
I wonder if part of the solution is what I was beginning to allude to previously, which is to somehow develop a legal framework where people just feel safe participating in research. Now, IRBs are dealing with everything in terms of their health and well-being, but part of it also is just feeling safe that their information, their medical information, their genomic information is going to be held tight. Nobody can re-identify them. Nobody could do surreptitious testing of them. You know, there's a lot of things, and I think that there are legislative things that could be built up to sort of create a culture to say it is safe to be a research participant in America. And I think part of that could be the IRB, but part of it is to build very strong protections, and there's some steps that have been taken so far in terms of discrimination against genomic information of an individual, but that needs to be strengthened. And I think if we could create a culture where people don't feel concerned about privacy, don't feel concerned about their information being used against them in some bad way, and if we have a more sort of an IRB system, that's maybe if it's more standardized, people would just understand it better and they would realize. I think all these protections would feed into greater participation, which is going to be key.
So I would just like to pick up on that theme and extend it a little bit further in terms of participation in clinical trials and sort of realistic clinical trials, bring up the issue about surrogate. This comes back to the IRBs. No 10 people on a single IRB will agree. No 10 IRBs will agree as to the appropriate endpoints. But part of the endpoints, you know, if it's death or some hard endpoint, is not realistic for clinical trials. So I think one of the issues that we have to deal with is, are there a set of acceptable surrogate markers that will shorten the clinical trial and that we can all agree on as a stopping point so that we can move these things forward but be confident that we are, you know, looking at the right endpoint. And I know that's a morass, but I'd like to just raise that and discuss it further.
Yeah. Well, you know, there is no process to do that. Okay, that's what I was talking about, the translational apparatus, science, evidentiary standards for how do you move a biomarker of some sort into using it as a surrogate endpoint has not been established. We have the Prentice criteria, which are highly unrealistic and have been around for a very long time, and nothing's ever achieved that. Even very successful things like viral load and HIV wouldn't meet the Prentice criteria. All right. So, but I have taken this up with academia and with the Institute of Medicine and so forth, and you know, there isn't scholarly effort behind this at the magnitude that could actually deliver. And so we're often faced with going to our advisory committees, which is just a certain group of people, asking them, because people have nominated different endpoints to us, what do you think about this one, and so forth. We are trying at the FDA to move this field along using our qualification process. We have a process where we let people come in with candidate biomarkers of different kinds. We try to qualify them for different uses, but that requires what are the bars? What are the evidentiary criteria that you have to get over? So we can't just make them up. We can't just say, oh, or not. I mean, we, the whole clinical community, we can't just say, oh, this endpoint is fine. There has to be some type of science underlying it, some process that we've agreed to, so that patients, practitioners, with confidence, if let's say a drug is approved using that, that it will deliver the benefit that is promised, at least most of the time. So that's another effort that I see is really lacking, is to try to understand what evidence would you use, how would you do that. There are standards sort of for clinical utility of various diagnostic tests, but here we're talking about pairing that in a way that allows, say, drug approval to occur, biologic approval. And it's a really interesting field that's been really under kind of powered, so to speak, from a scholarly point of view, in my opinion.
Yeah.
So, maybe the endpoints are critical and definitely would accelerate things, but maybe we can think about it a different way. And one way that I think about it, particularly because we're involved, we've just received acceptance for a paper that will describe a first-in-class drug against the GTPAs. And so I've been thinking a lot about this drug for the past year and a half, 2 years, as it's getting closer to publication and potential trials. I think the endpoints, without thinking of surrogacy in terms of death, et cetera, one can think of endpoints as embedding even the effectiveness or hitting the molecular target as an endpoint. And I would argue if I have my druthers about this drug that we've developed, basically, I would like to know whether the drug inhibits the target in the phase 1 trials. I don't want to wait to phase 2 trials or phase 3 trials to know that the target has been inhibited. Because although it is not a surrogate endpoint, as you both were talking about earlier, thinking of, well, if this biomarker is a certain level, then we know the patient will be cured or will live. I would think of it even more simply and more individually for every drug is that in the first trials, the first time the patient sees a new drug, we need to know whether that drug inhibits that target. Because if the answer is no, I, for one, whether I'm an investor, scientist, researcher, I don't want to proceed if that drug is not being inhibited. So,
About 10 years ago, we worked with the National Cancer Institute to have what we call phase 0 trial.
phase 0, yes.
For proof of concept, because what you're talking about, sorry about technical stuff, you consider that a pharmacodynamic endpoint. You're saying if the drug doesn't do what it's designed to do, why put effort into investigating it? So you should be able to get into people for that type of pharmacodynamic information. What we have found, or easily, but what we have found is that developing the assay is difficult, and is often that assay, that pharmacodynamic assay, is not developed parallel with the targeted candidate drug. And so get into people, or you do a phase 1 trial, and you don't know squat about whether it's actually doing what it was designed to do. And that's very unfortunate, I agree with you. We
Yeah.
still see that.
Just to come back on this issue of biomarkers and surrogate outcomes, it's very important. But the more we know, the more we believe we have a surrogate outcome. And so I'm going to return to the issue of registries, very careful registry, and careful clinical trials networks. They help you in another way. Once a drug is approved, you can monitor to see if it's really doing what you think it's doing and what complications there are. Everybody knows, you can do a trial on 1,000 people, not see a complication, but it could kill 80%, 10,000. You don't know that until after it's approved. But be that as it may, if you're early on the early side, have an excellent registry and excellent clinical trials network, you can do the testing you're talking about. In cystic fibrosis, we have 7 national resource centers that are dedicated, we have 2 of them here in Denver, and that are dedicated to looking at biomarkers to see how they will help us without
From an industry perspective, trying to develop sort of the hardly a reason to do it, right? Because drug gets approved, they thank you very much. So there's nothing actually that you can commercialize to help with, though. So certainly monitoring tests and monitoring pathway assay for progression of disease, that can be helpful. We are in fact working on some of those. But missed a very complex problem. I think maybe not suited for this discussion because it's so complex.
Yeah. I tell people all the time, you might not want me making a law about this.
Yeah. A lot of help with this.
Yeah, right. A lot that can let you do what you need to do.
We're talking about registries, we're talking more broadly about getting a larger portion of the population participating in research. It's not that we're just fantasizing about this. I mean, other countries are doing this, and other countries are doing some of these things better than us. And one of the concerns I have in thinking about the health and well-being of genomics here in the U.S. is a great concern that this, despite the fact that we've been world leaders in this for 25 years, we might lose that edge over the next 5 years. And that really concerns me.
Which, Eric, that's one reason why Chairman Upton and I are trying to look at this 21st century cures and trying to shift the national paradigm in how we're doing biomedical research and then taking it to the clinic. So tell us about some of the things that other countries, and we still have the edge. I mean, when I travel to other countries, they're still relying on their collaborations with our researchers and the wealth of knowledge that we have. But tell us about some of the things that other countries are doing that are helping them move upward, and what are some of the institutional barriers that we have in this country?
So, and I would start off by pointing out, I feel like there's a good number of other countries that their biomedical scientific enterprise are on their toes. They have great momentum. I hate to say it, but I think that right now, the U.S. biomedical research enterprise is on its heels.
Why?
It's for several reasons. We have to start with the funding trend. There are plenty of graphs that illustrate this, where other countries are investing upward. As you know, we've been flat at best, which means our purchasing power has dropped over the last 10 years. So immediately, that's a big component of it. But that's not the only aspect of it, because I mean, funding is clear. I think part of it is sort of also based on bold initiatives, and we're talking about some of these, bold initiatives, for example, let's take the largest capacity for doing genome sequencing right now, using technologies largely developed and supported in the United States, happens in China. And we shouldn't let that happen, but that's what's happening right now. The largest registries of patients with electronic health records and medical information and genomes being sequenced of individuals are taking place in a number of countries. England is the one most recently announced, bigger and bolder than anything we're doing here in the United States. And there's several other countries that are doing similar things, and there's several other countries I know of who are planning and will likely be announcing things. And so, and I think we've created a circumstance. I have 2 teenage children at home. One's just started college and wondering, should I go into science? And in the United States. And I think in some other countries you go to, and there's just this huge groundswell of excitement about being part of this revolution in biomedicine, because they're in a place where it seems to be valued, it's being rewarded. The U.S., the government, that country's government seems to be on the upward trajectory of supporting science. And it's hard for us to tell our college kids these days and high school kids that the trajectory is a healthy one for this. So, and part of it is money, but I think part of it is boldness. 25 years ago, when the U.S. Congress said, and the scientific community said, we're going to sequence the human genome, it was sort of like saying we're going to put a person on the moon. And I was very young when that happened. I remember it even as a kid this tall, but this was really exciting. We need boldness here. We need boldness, and we need a funding commitment and bold ideas that everybody can get behind.
Well, let me just say, because I was in Congress when we doubled the NIH budget in 2001, and that sort of led to that kind of excitement. We're gonna cure cancer. We're gonna find individualized medicine. And then, of course, what happened after and that was a bipartisan move. Don't forget, in 2001 when we did that, we had a Republican president, we had a Republican House, I think we had a Republican Senate, and we did it in a bipartisan way. And then, of course, over the next years, then it just languished. But then, the kind of the death knell was, in my opinion, the sequester, because not only did we have a flat funding for the NIH and everybody else. But then, every agency had to have these mandatory cuts. And so the first thing that got cut were grants going to the younger researchers. And that's what, you know, we've heard this. This is one of the, all of the themes we're talking about today are common themes that we've heard in these roundtables, but one of the common themes is that the younger researchers are discouraged from going into this field because they don't think, you know, they might have the best idea on some kind of cancer research, but if they're not gonna get if they're A, not gonna get funded in the first place, and B, not gonna have a continued funding source, why would they do that?
The other comment I made, and I think I've heard hints of it, but anybody can disagree with me. Many of us are roughly the same generation. When you double the NIH budget, there was a great warm feeling, this is wonderful, we're going in the